Okay, so hello everyone. You're all very welcome to the first SIMBA Lunchtime Seminar. This is the first in a seminar series that we've put together to just raise awareness about the SIMBA project and also raise awareness about some of our sister projects from the Microbiomes for Life cluster. For anyone who's unfamiliar with SIMBA, we're a now 5-year Horizon 2020 project. And the aim of the project really is to explore and investigate microbiomes and see how they can be used to improve food and nutrition security in Europe. So today we are going to have a series of flash presentations from some SIMBA partners and a special guest as well. And I'll ask that you hold all your questions until the end of the presentations. But if you think of something during the presentation and you want to ask it straight away, please feel free to ask it using the chat function, and we can pose it to the speaker at the end. As I mentioned, this meeting will be recorded today. It'll be shared on the SIMBA website and SIMBA social media channels as well. So I'm very pleased to introduce our speakers today. So first up, we will have Tamára Santos, who's joining us from Necton in Portugal. We'll also have Emilie Gullberg Jørgensen joining us from NMBU, the Norwegian University of Life Sciences. And I'm also delighted to announce that we have Davide Bulgarelli from the University of Dundee. Davide is joining us from the CIRCLES Project and is going to tell us a little bit more about his research and about the CIRCLES Project as well and what they're doing there. We had hoped to have a presentation from SIMBA Work Package 2 leader, Anna Maria Bevivino. However, unfortunately, she was unwell this morning and isn't able to join us, but hopefully we might get her to join us at a future seminar in the next little while. So I think without further ado, let's get started. And I will say that this is a lunchtime seminar, so please feel free to have your lunch, have a coffee, and just sit back and enjoy the presentations. So I think I'll hand over to Tamára. If you want to tell us a little bit about your research and what you're doing, we'd love to hear it. Thank you very much.
Thank you, Sarah. I'm going to share my screen.
Great, thank you.
So, hello everyone. My name is Tamára Santos, and today I'm going to present our work entitled "Microbiome Mediated Improvement of Industrially Produced Microalgae," the work performed by the companies Necton and MICROALGAE in Portugal. So, this work is integrated into the work package 3 of the SIMBA project. Marine microbiomes for a sustainable high-quality food production, more specifically to modulate and control microalgal biomass for a sustainable food production. So what are microalgae? Microalgae are unicellular microscopic eukaryotic organisms where most of them uses photosynthesis to produce organic matter and energy. And this ability of these microorganisms to have high contents of high-value products, like, for example, polyunsaturated fatty acids, pigments, lipids, proteins, and bioactive molecules, make them widely used in the biotechnology sector for food and feed production, as a supplementation in the health industry, and for the development of new cosmeceuticals. As for the microbiome, we are talking about the entire habitat, meaning the microbiota, that is the assemble of microorganisms belonging to different groups like bacteria, viruses, fungi, and archaea, their genomes, and also the surrounding environment. And when we join both of these groups, microalgae and the microbiome, it's important to focus on the possible interactions that you can have among them. So one of the most described interactions is where both groups of these organisms benefits from this association, like, for example, mutualism. So microalgae can produce compounds, like, for example, polysaccharides, dissolved organic matter, and oxygen that are taken up by the members of the microbiota that in return produces vitamins, antibiotics, and growth promoters. So we have an exchange of compounds between the two groups. So the co-culture of these organisms, if we only consider this synergetic approach, can be beneficial to both. In algal biotechnology, usually the most described group of microorganisms of the microbiota is bacteria, and they are often described as contaminants. But new reports show that indeed this co-culture can benefit the production of microalgae, not only by improving the productivity, but also the biomass quality and preventing parasitic or negative interaction of other microbiota groups. So this comes in line with the objective of this work, that is not only to characterize, but also to isolate and develop tailor-made microbiomes that can be added or co-inoculated with the microalgae, not only to enhance productivity, but also to increase quality of the produced biomass, increasing its market value. So our approach to start was to select microalgae species that were already produced industry and that had biotechnology potential. So we selected CIRCLES lutea, that is a marine haptophyte and highly used in aquaculture and cosmeceuticals, mainly due to their polyunsaturated fatty acids composition, omega-3. We selected Nanocoropsis oceanica, that is a marine orcophyte and is highly used for aquaculture, for feed. And also Curella vulgaris, that is a freshwater corophyte and used in the food sector, mainly because of its protein contents, high protein contents. The next step was to produce industry these species, and we evaluate also its nutritional and biochemical value of the biomass. So we use different production systems for microalgae, like open systems, like raceway ponds, and also closed systems, like tubular photobioreactors and flat panel photobioreactors. We used the volumes used were in a wide range, from 100 liters to 140 cubic meters. So from all the sampling points regarding the 3 species, we collected growth data to evaluate the productivity of the system and also performed the nutritional characterization for proteins, lipids, fatty acids, and also vitamins, focusing on vitamin B12. In parallel, for all these sampling points, we also established the bacterial isolates and also performed sampling for microbiome analysis. So following up the isolation and identification of the bacteria, we use culture-dependent methods, namely serial dilutions, where we obtained more than 1,000 isolates, but we selected the ones that were morphologically distinct. And we pursue with the identification of more than 500 different strains. For this, we can see that based on the relative abundances of the culture-bound isolates, we get a higher diversity for CIRCLES lutea with 24 families and 34 genera, and we emphasize the appearance of the group Hodobacteriaceae that is known to produce growth factors promoters. For Curella vulgaris, we have 14 families and 22 genera, with also the appearance of these 3 groups that have reported growth enhancing factors. And for Nanocoropsis was the less diverse, with 14 families and 17 genera. If we look closer to the similarity among species, as expected, we have a higher similarity among the isolates for the marine species, microalgae species, for Nanocoropsis and CIRCLES, when comparing the freshwater species, Curella vulgaris. We also found out some species that were already reported to be dominant in the interaction among microalgae and the microbiome. I want to highlight the Altera monodaceae, that is a genus known to be vitamin B12 promoters, and the other highlighted groups that are known to have growth factor promoters, not only in microalgae, but also in plants. This literature review also showed that some groups, like for example, Alomonas aquamarina, that was also isolated in our work, was capable not only to improve the productivity of the system, but also to improve the lipid production of the Nanocoropsis oceanica cultures. And this literature review was also important to select 100 species to proceed with the work. So what we did next was to select the species that were reported to have activities, and most of them belong to the group of alpha and gamma protobacteria that were known to be the dominant species in these kinds of interactions. And for these species, we started with the visualization of them, understanding how they grow, and after passing to the liquid media on non-selective mediums. So the bacteria belonging to the genera classifying as contributing to higher growth rates were put into growth for us to evaluate their ability to grow and afterwards to be added to the co-culture with the microalgae. At the same time, we also performed sampling for the microbiomes associated with industrial production for the same sampling points as we did for the isolation of bacteria and checking the productivity and nutritional value of the biomass to be able to correlate all of these different factors. And we try to not only to characterize the whole microbiome present in these samplings by high-speed centrifugation, but we also try to divide these fractions in two different parts. The phycosphere, that is the area immediately surrounding the phytoplankton cell that is known to be enriched in organic molecules, but also the supernatant, that is a fraction that contains the motile bacteria. We extracted more than 600 samples and we are performing the analysis now. This is also important because when we have the co-culture dependent methods, we only see a fraction of the bacteria that are able to grow in the medium that we selected. And when we do this approach of the microbiome analysis, we can see the complexity of the community and the differences among them. So the next steps include the analysis of the samples of the microbiome and also the growth of the isolated bacteria with the microalgae species to assess their effect. So this is all important because microbiomes are known to be essential for the microalgae growth. Some species cannot grow without the associated microbiota that they are present, and they also have benefits and impacts in not only their production, but also their quality. And this should be very helpful not to improve food nutrition, but also security of the microalgae production. Thank you for your attention. I'll be glad to answer any questions that you have, more specific questions. Thank you.
That was great, Tamára. Thank you very much. It's really interesting and just brilliant to hear more about what you're working on at the moment. I think we have a question actually from Emilie. Do you have your hand up?
No, I was just giving applause.
Ah, yeah, that's great. Thank you. And I don't know if anyone has any questions or if you'd like to add them into the chat, you can. No, not at the moment. I think Tamára, it was just great to hear more about the next steps with your research as well. So, and just in terms of, let's say, the time frame for that, so how is that going to play out within the project? I guess, does microalgae research happen very quickly or is it slow? It's hard. I'm not sure myself.
We have the production part was the more delayed part. And also the microbiome analysis will take some time, but we have all the data, so we just need to proceed on. All the growth experiments, because we are doing like a pre-test to check first their ability to grow, so we're gonna reduce even more the number of isolates, and only in the ones that have a good ability to grow and that show to be good for the improvement of the biomass, we will after add them in a factorial kind of experiment to assess their importance.
Okay, thank you very much. It's interesting, and I think we have another question from Renee as well. If you want to jump in.
Yep. Thank you. I have a question. Do you have any idea how stable the microbiomes are in your different cultures, especially in the open pond raceways, where you have, of course, interaction with the air and maybe a bird is pooping into it, and so how does the microbiomes change over time and under different conditions? Do you have any idea about that?
I cannot answer you based on the microbiome data because we only have the extracted DNA samples and we are still waiting for the results. But what I can say, that is not like final, but what I can say based on the isolation, that we know that is a method that only goes on part of the bacteria that is present in our culture is that based on, because I didn't explain here, but I did an experiment when I did the same production from the same inoculum in raceway ponds, in flat panels, and in photobioreactors that are closed systems. And I did it at the same time 3 times. And I isolated bacteria from this. And what I could see is even if I have a higher diversity in the closed systems that I was not expecting, because the open systems are much more likely to be prone to the outside factors, I have a higher load of bacteria. So what I think, that is not final, but is just an hypothesis, is that I have fewer groups that are able to stand this kind of rough conditions, but in more quantity, meaning more dominant. But it's just an idea. I need the microbiome data to confirm this hypothesis.
Thank you very much.
You're welcome.
Great, thank you. And I think we have one more question in the chat from Sylvia, and she's wondering, which culture medium do you use to isolate bacteria?
So I used non-specific mediums. I used PCA, plate count agar. I use marine agar, and I use TSA, tripsoy agar.
That's great, thank you. I think we have time for maybe 1 or 2 more questions before we move on. I don't know if anyone would like to ask anything in the chat or by raising their hand. No, it doesn't look like it. We might have some time at the very end of the seminar if anyone would like to ask any questions then, or you can ask Tamára questions directly in the chat as well. So Tamára, thank you very much for that. It was really interesting to hear more about what you're working on. It was great to have you. So if we move on, thank you. I'm going to invite Emilie to join us now and share her screen and tell us a little bit more about what she's working on at the moment.
Yes, let me see. So now you see my presentation?
Yeah, that's perfect.
Yeah, that's great. Hi, everyone. My name is Emilie Jørgensen. Unfortunately, none of my NMBU colleagues could join me today, so hopefully I will be able to answer any questions you might have at the end. I will talk a bit about the NMBU contribution to the SIMBA project. Yeah, so a bit about the contents of my presentation. I will go into and introduce NMBU's role in the SIMBA project, a bit about the tasks in work package 4, which we are mainly involved in, some analysis setup, and the in vitro digestion method, which we use, some preliminary results, and then conclusions and future research at the end. So about NMBU's role in the SIMBA, we see that there is an increased demand for plant-based food or plant-based products, and a need to develop new and nutritional foods. And the use of microbial fermentation can be like a good tool or a new tool to produce novel foods from plant-based raw materials. And these novel foods need to have a high nutritional quality, which is NMBU's role to look at in the task 4.3, which we collaborate with our partner Luke in Finland. So a bit about the tasks. For task 4.2, conversion of plant raw materials and side streams by microbial processing, is where we look at like fermentation of plant-based raw materials. And task 4.3, which is like the main task we are contributing to, is the nutritional quality and quality assessment. And then task 4.4, which is analysis of raw materials, side streams, and effect of fermentation and digestion on the composition of the foods. So in task 4.2, we have had a master thesis on a fermentation of pea and faba bean flours, where these master students look at the effect of fermentation on the pH levels, organic acids, carbohydrates, content of phytic acid and FODMAPs, and free amino acids. And what remains for us in this task is to report the results. The results are obviously in the master thesis, but we are also working on an article which will be supplemented with results from other projects which we have been working on at NMBU, like outside SIMBA, but also with plant-based raw materials. And then in task 4.3, we look into how we can evaluate the nutritional quality of foods from task 4.2, so fermented plant-based materials. To do this, we look at digestion or digestibility of the products, and so far we have used an in vitro digestion model, and this is to get an idea of the digestibility and is a good measure before we do in vivo analysis. We also look at the chemical and physical analysis of the food before and after processing, or before and after fermentation, and then after in vitro digestion. We look at the increase in nutritional compounds, bioavailability of nutrients, and the decrease of anti-nutrients. So what we have done so far is looking at obviously the master thesis from task 4.2, but also we have collected samples from partners. Our partner Luke in Finland has fermented some samples, which they sent to us to optimize the methodology. We prepared the protocols for the in vitro digestion, and we have digested samples from pea, lentil, and faba beans. And we have also looked at the microbial survival during both the fermentation and during the in vitro digestion. So what remains to do in this task is to analyze the samples before and after digestions and a report of these results. So task 4.4, which is validation of the methods, which is kind of what remains of task 4.3, where we look at samples before and after digestion. We look at the protein degradation after digestion and fermentation, increase of beneficial compounds such as phenolics, flavonoids, non-starchy carbohydrates, soluble and non-soluble fiber, vitamins B12 and K. And we also look at the decrease of anti-nutritional compounds such as protease inhibitors, phytic acids, FODMAPs, glucosinolates, vicin, and convicin. So a bit about the analysis setup. We have the fermented samples, which Luke have kindly sent us. And then in the yellow bubbles is kind of what we have done already, which is the in vitro digestion. We sampled 3 different points of the digestion, after the gastric phase and after the duodenal phase in 2 points. And then we have looked at the bacterial survival during the digestion. So what remains to be done is to analyze the fermented samples both before and after digestion. And a bit about the in vitro digestion model. This is adapted from the InfoGest digestion model, where it's split into the oral phase, gastric phase, and the duodenal phase, where we, like I said, sampled from after the gastric phase and during 2 time points in the duodenal phase. So some preliminary results. I can't really show you our results just yet, but I can say something about what we have discovered with the results we have. So the fermentation with some of the selected strains did significantly reduce the contents of the anti-nutrients phytic acid and FODMAPs, which is a good thing. Phytic acid can reduce intake of protein and minerals in the digestion, and FODMAPs can cause discomfort during digestion. So these are things that we want to reduce. And some of the selected strains did also survive the in vitro digestion. which is good if these strains could possibly be probiotic bacteria. And it also found that the plant-based food matrix improved the bacterial survival. Without the food matrix, like just the strains, we found that the strains didn't really survive the digestion, but within the food matrix, there was a higher rate of survival. So the factors that can impact the bacterial viability could be the food composition, the food processing or the fermentation and the storage of the food, the probiotic food matrix interaction, and also strain-dependent characteristics such as pH and bile salt tolerance, which is something we will look more at. So what this tells us is that the fermentation showed reduction of anti-nutrient content and also improvement of digestibility, which could mean that these products could, or this processing could improve the nutritional quality of the food. And the in vitro digestion did show that some strains survive the pH and bile salt tests and the digestion, and also that the food matrix protects the bacteria, which again could tell us that these fermented products are effective as probiotic bacteria or probiotic food products. And here's some graph. The graph on the left shows the pH trend during the in vitro digestion. So as you can see in the gastric phase, the pH drops to about 3, which is where we also expect to see bacterial or not the bacteria not to survive this low pH. But on the right, we have some graphs from bacterial survival during the digestion in fermented faba beans. And you can see that not only does the bacteria survive the gastric phase, but in the duodenal phase, it also seems to grow the bacteria. So this could mean that the food matrix actually protects the bacteria and helps them to thrive in the digestion. So to conclude from this, we can say that there's a bright future for microbiome applications in sustainable food production, and fermentation with selected isolates improves the nutritional quality of plant-based foods. So in the future, this could mean that we want to obtain more data of the improved nutritional quality during fermentation, and we want to find out more about the mechanisms behind this, both during and after the SIMBA project. And after learning more about this, we could produce new and more nutritional foods. And in like a bigger picture, kind of, we think this is a good way to reach the Sustainable Development Goals 2 and 3 to eradicate hunger and ensure good health and well-being for all. So yeah, that is everything. Thank you so much for having me.
That's great, Emilie. Thank you very much. It was really interesting to hear more about the research going on in NMBU. I don't know if anyone has any questions for Emilie. And like Marie, is your hand up?
No, sorry, that was just clapping.
I can't tell the difference. Yeah, thank you very much. Yeah, David, I hope that's a hand up and not.
Yeah, it's on purpose this hands up. I have a question for Emilie. So first, thank you for your very interesting presentation. My question is, where do you source your raw material? So, for instance, the faba beans that then you subject to fermentation. Is this a European Union product? It comes from the international market. Can you comment on that?
Thank you for your question. I'm not entirely sure because, like I said, we receive this fermented foods from our partner Luke. So I would assume that they use these raw materials that they have, but I'm maybe knows more about this than me, but we have collaborated on to find which strains to use, and then they do the fermentation and send it to us. So yeah, sorry I couldn't answer better, but yeah.
That's great. That was just a curiosity.
Okay. And we have another question from Sylvia in the chat. So did you correct the pH during digestion?
Yes, so before we do the digestion, we do a test digestion where we adjust the pH in all the samples. And then for the actual digestion test or analysis, we have recorded how much acid or base we need to use to correct the pH. Because obviously to measure the bacterial survival, it has to be, we can't measure the pH during this. So we measure it beforehand, and then we know how much to adjust with when we do the analysis.
I don't know if anyone else has any more questions. No, I don't think so. And as I said, we might have some time for more questions if there are any in the chat as we conclude our presentations, or you can feel free to message Emilie separately if you have anything to ask her. I think there's increasing awareness about, or increasing popularity about, these plant-based diets as well, and people are really trying to find out more about them. So I think it's a really interesting space that your research is in, and it's great to hear more. So thank you very much for that. So what I think we'll keep moving. We're kind of coming into the last half hour of our seminar, so we'll move on. And I'd like to introduce Davide Bulgarelli, who joins us from the University of Dundee. And as I mentioned before, he is a member of the CIRCLES Project, which is one of SIMBA's sister projects in the Microbiomes for Life cluster. So Davide, thank you very much for joining us today. If you'd like to share your screen, you can go ahead.
Okay, so let me just 1 second sharing my screen.
Yeah,
okay.
Take your time.
Can you see my presentation now? So the first slide?
Yeah, and it's in presenter mode as well. Yeah, so
okay, fantastic. So, well, first of all, thank you very much for this kind invitation. So thank you, Sarah, for having organized this this first seminar series. So I think it's a very good idea having this type of let's say informal networking among the microbiome projects, because of course we are moving in the same direction, and will be very useful to build upon our experience to essentially avoid mistakes or duplicating things that perhaps we have proven are not working. So very briefly, CIRCLES is the ethos of CIRCLES is the very same of SIMBA. So capitalize on the microbiome across food chain to essentially provide a better, more nutritious access to food for Europeans and beyond. So in today's presentation, I'm going to give you a very brief summary of the project, emphasizing on what we are doing as a University of Dundee and essentially as a working package 3, the one that I'm coordinating in the project, following the indication I received on the email. So there are these different points that we were asked to essentially touch upon, and I will stick to that, and I will do that throughout my presentation, obviously, and hopefully, sorry, I hope that this will trigger a discussion later on. Okay, so in the working package 3 of CIRCLES, we are interested in plants. And plants are growing in soil, where they define different theater of activity. In particular, the interface between root and soil is of interest for the interaction between a multicellular organism like plant and the microbes that are present in soil. So soil is one of the most diverse microbial ecosystems in the entire planet. And these type of interaction are not, let's say, they have consequences for the plant. And similar to what Tamára said, so we need to define the terminology right? So the microbiota refers to the community of microorganisms that encompasses bacteria, fungi, nematodes, protozoa, viruses, you name it. Predominantly bacteria are perhaps the best characterized or more abundant anyway in this environment interacting with plants. Now, when you take into consideration the surrounding environment, which is the thin layer of soil surrounding the roots, defined as rhizosphere, which is modulated by the plants. The root itself, so the inner part of the plants, and even the background tissue that may represent a site for microbial proliferation. So you define the theater of activity of those microbes, and therefore the microbiome and the different microbiomes. And of course, for comparison, we have what agricultural scientists often refer to as bulk soil or unplanted soil, basically the soil without the plants. This type of comparison helps you to identify what is the impact of the plants on these communities. And considering especially that the interface between root and soil is rhizosphere, is a site for resource exchange. Plants are autotrophic organisms, but they require minerals to grow, and those minerals are coming very often from soil. So there is a site of nutrient acquisition, and yet you have a community of microorganisms that are, let's say, modulating or contributing to these processes. I hope that you agree with me that the way we look at plants is fairly similar to the way medical scientists look at the microbial communities inhabiting our own digestive tract. And in analogy with the gut microbiota of vertebrates, plants do have their probiotics. Those are called plant growth promoting rhizobacteria or microorganisms in general, if we're referring beyond bacteria. And essentially, microbes, mostly derived from soil, can do a number of things for plants. For instance, they facilitate the acquisition of minerals from portion of soil that plants may not be able to access them. They can protect plants from parasites, so fending off potential invaders, and also contribute to what agronomists refer as a biotic stress, so environmental conditions that are undermining the fertility of soil, drought, salinity. So there is a number of things that those microorganisms can do in return from essentially what plants can provide them via photosynthesis. It is a mutual exchange. However, the interaction between plant and microbes in soil in particular may go terribly wrong. So there are a category of microorganisms that are part of the microbiota that are particularly greedy. So they take from plants and they give back nothing. And then they cause what, again, in analogy for what's going on in the gut microbiota, is a dysbiosis. They become dominant and plants suffer from them. And the consequence of that, you can see in this picture, is essentially a loss of yield and a loss of harvest for farmers. Therefore, I hope you understand, I hope you agree with me that understanding the fine relationship between plant and microbes, in particular in soil, is critical to ensure the optimum growth of the plant and ultimately sustainable crop production. And on this concept, the work we are performing in CIRCLES is essentially carved. So in CIRCLES, we are looking at multiple food chains. In the working package 3 that I'm reporting today, we are looking at the plant food chain, and we have essentially two models. One is spinach, and the other one is tomato. And within the project, we have other food chain, for instance, farmed animals, farmed fish, let's say fish in the Atlantic. And the concept and the approach we are following for each one of these 7 food chains is essentially modular and similar. So what we consider the trademark, or anyway, a key element of the CIRCLES project, is what we have defined as lab in the field. So this is a real participatory approach of stakeholders. So, for instance, in the case of the working package 3 and the plant, we deal with farmers in the field. So their field are our labs. So we are characterizing and studying plant microbiota interaction in that condition. And we benefit from that because, of course, farmers and other stakeholders have decades of experience, and they contribute actively to our experimental design. So it's not a bottom-up approach. It's a more, as I said, a participatory approach. And again, this applies to all the food chains we have in the project. The core of the work is what we have defined as an observation phase. So we are learning by nature. We investigate the composition of the microbiota, in our case of spinach and tomato in that farm, and we try to infer, or we try to define what makes the spinach and the tomato microbiota distinct. And we are doing so also by looking at what are the accessory of the crop production system. So for instance, when you grow a plant in the field, your ultimate goal is getting an harvest out of that. But there are some residuals in the production of this crop. For instance, water, or for instance, the machinery that are used in the field. Those may be at the same time a driver for microbial diversity and a reservoir for microbial diversity. So if we are interested in the dynamics, we need to have them into our experimental design. And then the last phase is the so-called intervention phase. So after we have learned how or what makes the plant microbiota for our two models distinct, so we try to manipulate key members and try to see whether this is a beneficial impact on the essentially on the productivity of the plant and on let's say the sustainability of the crop production by manipulating directly the microorganism or by manipulating elements that in turn can impact on the microbiota. And while doing so, and again following what is the ethos of the lab in the field, we maintain a very active communication channel with the general public. So the idea is that whatever we are going to do or we are going to propose shouldn't be top down or parachuted to what are potential consumer, but again, they need to be informed of what are the modifications we believe will increase the sustainability of food production in plants and any other of the 7 food chains that will be part of the project. Now, how we get along with that? Essentially, our lab in the field are in Italy. So in there, we have several farms. We have established several contacts with farms in Italy. And that's as I mentioned, the observation phase is essentially going and collecting samples in this field. And once this is done, everything is shipped back to Dundee, which is a city in the north of Scotland, where our labs are processing the material acquired in our labs in the field, and essentially we reconstruct the microbiota of the plants using what scientists refer to as cultivation-independent approaches. So we use the DNA as a proxy for instance, or to elaborate a census of the microbial communities that are populating our plants, what kind of function they may provide to the plant, and essentially what are the drivers of this community if we look at the so-called covariates, for instance, different farms, different season of farming, different destination of the final product, and so on and so forth. So once we have acquired this information, we are in the position to try to manipulate them for the benefit of agriculture. And as I say, the manipulation, at least in the economy of circles, will operate using inoculants or using industrial derivatives that may shift microbial composition. And what we are trying to do in circles, and for what is concerning my lab beyond circles, is progressively moving from the left-hand side of this picture, which represent the, if you want, the traditional way of agricultural production, which is deeply rooted in the fossil fuel economy, to something that is less dependent on that, and of course less reliant on that, and more reliant on renewable sources. So our idea is really try to decouple sustainable crop production from the fossil fuel economy. And while a month ago, a bit more than a month ago, this was essentially a useful alternative to reduce the footprint, the environmental footprint of agricultural production, the dramatic events that have unfolded in the past months are also a stark reminder that we can't take fossil fuel economy for granted. So now this is not anymore an option, it's becoming a reality. Okay, so what we have been doing in these years in circles, despite the let's say the disruption caused by the pandemic, let me remind you, our lab in the fields have this tight link with the environment. But for instance, if during a pandemic we couldn't access the fields, we lose an entire harvest. And the same was true for the possibility of processing the material in the lab. So there were delays, but we were able to acquire our sample. And now essentially similar to what Tamára was saying, so we are really pushing hard on the sequencing phase to try to gather more data and try to really interpret this data, and ultimately then apply this knowledge for the intervention. And essentially, this is where we are going to go in the, I would say, upcoming months. So we were particularly pleased for the sampling we have performed. So I think that we have the raw data to generate the knowledge of the microbiota of our crops. The more the merrier, in the sense that with more sequencing we'll get more data. And now it's as mentioned in the previous slide, so there is a sense of urgency. Some of the approaches that farmers were using, for instance, in the past campaign, may not be so readily available in this campaign. So alternatives, as I mentioned, are becoming a real necessity for them. And we are really try to deal with that and try to optimize this also in our project. And that's pretty much all from my side. So I would like to thank my co-workers at the University of Dundee and the partners within CIRCLES that have contributed the most to this spinach and tomato line of investigation. And of course, all you for the kind attention, and I will be very happy to take any questions.
Davide, thank you very much for that. It was very interesting to hear more about your research and actually the CIRCLES project as well. And I think it's great that we can have this collaboration. You know, we hear a lot about the projects, kind of what's going on, but in terms of the details of it, it's great to dive in there with that with you. Thank you very much. And I don't know, does anyone have any questions for Davide at all? Yeah, Annika?
Yes, hello. I was wondering, maybe it's an odd question, but are you also working with simulation models to simulate the effect of the microbials on the plant?
So is a question, do we work with models? Not yet, in the sense that we are still in the phase of acquiring the data. Obviously, this could be an interesting avenue in terms of manipulation, to try to identify perhaps hubs within the microbiota of the plant that could be manipulated in a more efficient way. So we haven't went that far as now. So I should have also mentioned that beside the individual aspects of the analytical part and the fact that we may interpret our sequencing data, let's say the plant sequencing data within the plant food chain working package, there is a dedicated working package that will collate all the sequencing data in terms of interpretation, and they are also exploring multiple avenues to identify determinants and key members of the microbiota. So there will be this part which is perhaps beyond what is the scope of our individual working package.
Thank you.
Great. I don't know if anyone else has any questions. You can feel free to ask them in the chat, or you can raise your hand. Davide, I thought it was particularly interesting when you were discussing how you're involving the public, you know, in your research as well. I think that's really important, that you're creating a pull from them rather than just kind of pushing the final product nearly onto them. I think it's a great way to approach this type of work, because ultimately it's going to be them eating the crops that are produced.
This is a strategic objective of the project, and yeah, involved essentially each one of the let's say food chain has its own outreach activity, or try to engage with stakeholders in multiple ways, or the general public in multiple ways. We strongly believe this is a fundamental element for the success of the project.
Yeah, no, I would agree. I think it's very interesting and a great approach for the work as well. I don't know if anyone has any other questions. No, I think you're safe, Davide. There's no more there. Thank you very much for the presentation, and as I said, it was great to hear more from CIRCLES and have your input during this seminar. I believe if anyone has any more questions from those speakers, feel free to put them in the chat or raise your hand now. But if not, I think we'll conclude our seminar. I just want to say thank you to everybody for attending today. This is the first in our seminar series. We're hoping to have one every quarter now for the rest of the year, so that we can raise awareness about the work being done as part of the SIMBA project, and hopefully we'll have some input from some of our other sister projects too. So thank you for everyone for your time today. And if you have any questions for the speakers afterwards, you can feel free to contact me as well. So thank you everyone, and it was great to have you here today. So thank you very much. Bye now.